Hello.
Hello, good morning.
Hello. We're gonna wait a couple of more minutes and then we're gonna go live on YouTube. I hope everything will work out. Hello, Sandrine. Hello.
Hello.
Hi.
We'll wait like 1 more minute, 2 more minutes, and then we'll go live on YouTube and I'll start because I'm not only hosting today but also presenting. So if something goes wrong, don't go too hard on me. We can't go live on YouTube. Sorry for the delay now.
Don't worry, it will be okay.
Yeah, I'll send you the paper already into the chat. And maybe I can just start because, hey, Annie, maybe I can just start and then afterwards it will be uploaded to YouTube. So let me share my screen as well. We will have an exciting paper to look at and to discuss. Which was published in Nature this year, earlier in May. It's, the title goes Learnable Latent Embeddings for Joint Behavioural and Neural Analysis. I hope you all found it. I hope you can all access the chat. So, it's by Stefan Schneider, who is a PhD student, Jin-Fa Lee, and supervised by Mackenzie Mathis. And the global summary of what they basically did was tackling one of the fundamental goals of neuroscience, and to map neural activity to behavior, and basically to model neural dynamics during adaptive behaviors, to probe neural representations. So they explored with using deep learning and another algorithm. They explored hidden latent dynamics in the brain and found really cool results. So just like around about the background. So what neural latent embeddings can do, what we also work with quite often, is that they can reveal underlying correlates of behavior, but there are still currently problems with doing this, for example, explicitly and like flexibly, especially on neural data. And also, for example, to leverage joint behavior. That's what Mackenzie Mathis is quite famous for, because she's been working quite a lot on joint behavior. And there are also other problems with basically with those learnable latent embeddings. For example, we're still yet limited in the number of neurons and the length of time that we can record from, for example, an animal session. And other methods such as, they discuss other methods such as PCA and UMAP that we also work with quite often. And they say that PCA, for example, comes at cost of performance, and UMAP is, while being a good nonlinear method, they lack, for example, ability to include time information. And this time information is oftentimes given in neural recordings, and it could be just cool to embed that. So they wanted to basically disentangle what is actually giving rise to behavior. And so they created this new method called CEBRA. I can't even tell what's what it stands for. They have it somewhere here. We'll look it up later. So I'll walk you through the method first, and then through one of the applications, or one of the things that they showed was quite cool. So this CEBRA method is basically combining nonlinear representation learning and self-supervised learning. And the result of it is a dimensionality reduction method that can be jointly applied to behavior and to neural recordings. So it can basically generate those embeddings that we'll get into a little bit later across, first of all, multiple subjects. And it can cope with distribution shifts between experimental sessions and subjects, and also recording modalities. So that's basically their core thing, and a very, very cool result. So let's get into it a little bit deeper. to first walk you through the process, how this algorithm or how this method works, is so in core, it's an application of a nonlinear ICA, which is independent component analysis, on constructive contrastive learning. So first of all, we need data, of course. And here we have behavioral labels. No, I can't. Unfortunately, I can't highlight it here, but we have behavioral labels, we have time labels, and we have neural data. And the contrastive learning part is happening a little bit later, but yeah, it's basically, to just give you a little bit of background, it's basically a technique that leverages contrasting samples. So we have positive and negative samples, for example, against each other, and then it finds attributes that are common, and then it finds also attributes that basically can separate those samples. So and that's basically the next step here to determine this in those pairs of behavioral labels and time labels and like all the data that we're collecting. So they use afterwards that, like after that has been decoded, they use a nonlinear encoder, right? Which is here. Here they just use a CNN, a convolutional neural network, to train the model on those contrastive learning objective. And what's happening here is basically what I just described. And from this encoder, basically features are generated in the final layer that lead or can construct this low dimensional embedding space. So I hope that is okay. There are people joining. I'm sorry. It's a little bit difficult to coordinate both things. So basically we get from this encoder features, we get a low dimensional embedding of the data that we put in. And what they basically modified here, or what is like the very core of their work, in my opinion, and I think they also state it quite often, is that they modified this loss function for contrastive learning. And then they introduced a new modified loss function for it that allows to basically sample from discrete and continuous data streams to build this representation space that is highly accurate, which I will then demonstrate in a bit. So they first took synthetic data, because that's how you first test your idea and test your model. And these are basically each point, like each little part here, is the spiking rate of 100 neurons, and they reconstructed this true latent space basically with their CEBRA algorithm. And when you look at the R-squared, just the evaluation score, the reconstruction score is pretty, like, one of the, first of all, the highest, the median is the highest. And you can see here, there's a method that actually yields also higher scores, but that's a, like, if you look at the distribution, it's highly distributed, like it's not really consistent. So here, the CEBRA algorithm is way more consistent. So how they basically applied it to a little bit, well, easier application was that they looked at the positional decoding that the CEBRA algorithm can do in the rat hippocampus. They had data from electrophysiological recordings from a task where the rat has basically a track which is 1.60 meters, and it can like go just to rightwards or leftwards on this linear track. And they had the neural data and the behavioral data, so you could observe basically right where the rat would go. And they applied their algorithm to that. Let me show you a really cool video. It's in the supplementary information. I hope you can all see it. So we have the true position of the rat in centimeters, right? And then we have the CEBRA algorithm that is basically encoding it from the neuronal data as well. And if I play it one more time, can you follow the little dot here? This is basically the latent space embedding. And you can see that this circle basically here in the video is a smooth embedding that reveals the position of the animal on the tracks, because you have like the color coding is also, you can see that it's color coded, right? Where whether the rat goes to the right or to the left, and how much of a distance it is. So there is basically a continuous loop, as you can see, of neural latent activity around the track. And yeah, this is quite cool, because it's basically a high performance decoding algorithm that also ensures consistent embedding. I'll show you why it's consistent. Sorry, here we go. So they tested also other algorithms against each other with the same task, with the same data. And you can see here in figure E, the comparison basically of how consistent those algorithms are. And with the CEBRA method, you see a consistency of like it's overlapping almost every time to more than 95% in comparison, for example, to the UMAP. And here, to make it like more comparable, they reduce the dimension of the CEBRA of that method basically to 3 dimensions, and in UMAP to 2 dimensions, for example. But the CEBRA algorithm can also go up in the dimensions, so it can also go up to, I think, 128 or something. Is even better in that case, but yeah, it wouldn't have been comparable otherwise. So I hope that was more or less, understandable. I need to keep track of the time a little bit as well. So what else they also showed, that the method can be applied across sessions and for multiple animals, on the example of this four-lip movement behavior in a primate. I won't go into that too much. Basically, what they did here was that they recorded from area 2 of the primary sensory cortex of the macaque, and they discovered latent dynamics during an active motor task where the macaque was like moving a joystick. So yeah, that's like another thing I won't go into, but what I will demonstrate, like another super exciting application of what they did, was that they encoded complex dynamic visual information from the V1 of the mouse. And it was basically, they were able to see through the eyes of a mouse and reconstruct, based on the neural recordings, what a mouse was seeing during a movie, like a natural full movie. So this is just background, or more or less background information. So they trained like it's on Neuropixels and also on calcium imaging, but this is a little bit outside of the scope right now. What I will show is sorry again for the scrolling. What I will discuss with you is oh wow, it's not as long. They have like just a lot of supplementary material here. So this is the decoding of natural video features from the mouse visual cortical areas, and that's basically a more complex application of this CEBRA method. So what they basically did here was they took neural data from the visual cortex of many mice, and then they trained a deep learning model. They trained the CNN, basically, that I discussed earlier, to reconstruct or to decode what the animal is actually or was actually seeing. So the algorithm here can use, okay, that was in the other figure, but it can use basically a frame-by-frame video feature as label. So it has like every frame of a video has basically a label of multiple sensory input to then extract this neural latent space of visual cortex, basically, when a mouse is watching a movie. So they used here, all the data that they used was open source. So for the macaques, they used the data provided by a lab here. For the mice data and rat data, and they used the Allen Institute data. And what they did was, so first of all, they had basically two modalities: electrophysiological signals of the mice, and there's also a quite new method called it's not really super relevant, but it's called two-photon casein imaging. And it's quite cool because they use genetically engineered mice so that if their neurons are firing, they glow green. So they could just record it from the camera, which is quite cool and non-invasive. So what they did was basically taking many mice recorded basically the neural data of many mice watching a movie, and use that as an input for the training of a deep learning model. And then they took those mice, or they took a new mouse which hasn't seen the movie before, and then run its data through the algorithm that was trained through the model. And the model was basically predicting what the mouse was seeing frame by frame. And the super cool thing is that the accuracy here was greater than 99% in the decoding. And I will show you, actually, before I go back to the figure, I will show you again a super cool video where you can see that applied. So the CEBRA network basically applied. So you have the two-photon recording, you have the Neuropixels recording, right? And earlier we saw like this CEBRA embedded space. It was quite easy, basically, right? It was like two circles, more or less, because earlier it was the example of a rat just moving rightwards or leftwards, and here it's way more complex. But you can see the true video of the movie and then the predicted video. The predicted video is lagging a little bit, of course, but it is super accurate, like quite accurate to 95%, which is super cool, I think. So and that's basically how the embedded space looks like, where basically each of the points is decoding a certain frame. All right, so if we go back to the decoding of neural video features from mouse visual cortical areas. So we can see, like, that's what we just looked at, right? We saw the embedded space, we saw the original, and then the original frame. And here they also they used the nearest neighbors decoding on CEBRA, right? So basically the predicted frame, which is exactly the same, and also they compared basically to a nearest neighbor baseline and bias baseline. And you can, well, I'm not sure if you can actually see it that well. Maybe you can zoom in. So you can see here basically that the decoding accuracy is the highest here with the CEBRA algorithm, and here basically as well. So right, so here's the frame classification, the scene classification. They show it's the highest with the CEBRA single frame performance. It's basically matching the ground truth. And what they wanted to look into as well is what cortical area is actually, where do they get the most reliable signals from? And they found out, or they saw that it's the V1 has the highest performance. So the decoding data from there has the highest performance. And they also looked even more deeper into the decoding by layers, and saw that layer 2 and 3 and 5 and 6 perform significantly better in comparison to layer 4. So I think my time is over, so we have a little bit more time for discussion as well and for questions. But I think it's like quite exciting to see that basically the representation space is rich enough to understand even this level of complexity and resolution, like frame by frame of a movie. And yeah, we could maybe think together about potential real life applications, and also, yeah, they discuss, for example, one application for humans to restore vision, which yeah, we could maybe discuss. Thank you very much. I'm not sure if I can stop sharing, or if I should stop sharing. Maybe not, but I can already see hands rising. Amazing. I think Michelle was first. Feel free to unmute yourself and fire away.
Thank you. I got two quick questions. One is when they compare the CEBRA to the other models, I noticed that they actually used 3 dimensions, and in the other models they use 2 dimensions.
Yeah.
And so like the argument is like there is more variance explained or more accuracy with CEBRA, but if you increase the number of dimensions in your model, you always have more explained variance in your model. So I can...
That is true. But these are so first of all, these are the models that are comparable or can do the tasks that CEBRA's objective was as well. And also what they did was, for example, with this conv, I'm pronouncing it wrong, but conv PVAIE, they did something that actually improved that model, so it made it more comparable.
My point being, they could have did UMAP with 3 dimensions in order to compare it, which would have been more comparable.
Right.
It's like if you do a principal component analysis and you just show 2 components, you're going to explain less variance than if you do 3 components.
Yeah, I see. I don't know why they didn't do it with the UMAP, why they didn't use 3 dimensions. But yeah.
That's weird. And the other thing is about the video prediction. Are they predicting the frame or the content of the frame?
The frame.
The frame. So it's not reconstitution exactly of what the mice are seeing. It's like a statistical prediction of the most likely frame of the video that everybody's seeing that is done here. And that's why it's lagging, but it's neat. It's different from the framework of people that are trying to predict what humans are seeing from fMRI, for example, which is, you kind of see what's going on, with sort of messy. There was just a little clarification.
Yeah, I think they used the frames to train the model, so that's why the model is also predicting those frames.
But and then you predict a frame, but if you were to predict like out of sample videos, where we don't have this information, they're not showing it.
Right. But
okay.
I think so. I hope I'm not saying anything wrong, but I think so.
Okay.
Yeah.
All right. That's all from me. Thank you.
Sure.
Yeah, I mean, I had the same question, but yeah, I also didn't understand about the prediction part, but now, yeah, I mean, if, for example, we want to predict a new video, we don't know what mice see, right? So
right.
So it's just, yeah. Okay. No, I mean, it was basically those questions. So yeah, I don't... maybe the question then, why do we need this method? What like exactly we can do with this method? Like, I mean, I understand why we need, for example, UMAP, but I don't understand why do we need to embed the time. Are they talking about it, like time dimension?
Well, yeah, that's basically the neural recording, right? Like that's the time dimension that's almost available. Yeah, always available in neural recordings. And usually, so that's, for example, an advantage in comparison to the UMAP and to the other algorithms that that can't embed the time component into their...
yeah. The question is, why do we need to embed time component?
Well, because I guess like the neural dynamics are also linked to the time, like when neurons fire, it's also a time component that is in there, right? Michelle?
Time is one
Have an
dimensional,
answer?
so I don't think, I mean, if, for example, in my idea, I don't know, maybe I don't understand. So if time has like multi-dimensional thing, then we want to reduce it to one latent component, time latent component, and use it for something. But if we have just linear milliseconds or seconds, I don't understand, where do you compress it? I don't know, maybe I don't get the idea of this method then.
And yeah, that's something that I didn't get when they say, oh, UMAP cannot like compress time or like use time. But you know, in the end, it's just a matrix of numbers that you enter in those algorithms. And you can totally have time as one variable in there. UMAP will compress it.
Yeah, but not a jointly used. So they are combining, you know, behavior and neural recordings. So you have, yeah, I think that's basically what they're stressing, that they can jointly combine those things. We can individually, we can do neural recordings and compress that into lower dimensions, and also behavioral recordings and compress that. But like, yeah, to get a latent embedding space that is learnable, that is also consistent. For UMAP is, for example, not consistent, right?
It is quite consistent.
But not over, so we have here, not over individuals. Look at that, for example. So if you do the same neural recording for the same task of different rats, the consistency here is 30% in comparison to the CEBRA algorithm that is over 90%.
I see what you mean. It's just like the argument of not being able to put the time, because you can completely combine behavior and neuronal recording in your map. Just gotta put it in this matrix.
Maybe one of the features of you, I don't know if that's, I have to double check, but maybe you cannot use two variables that highly depend on each other. So time and neural recording, they will, I don't know, highly depend on each other. No, that's why maybe it kind of, yeah, I don't know.
I don't know. But the result looks statistically better, although they have an extra dimension.
Yeah, I'm wondering why they didn't add that to UMAP, actually.
It is strange.
That is strange.
Maybe in supplementary materials they do.
Yeah, they have a lot of supplementary materials.
Okay.
But yeah, they did quite a lot, and they also filed a patent for it. So they're like doing great with what they invented. Maybe my me walking you through that paper wasn't too clear, but yeah, feel free to open it up and read it yourself, and scroll through all the supplementaries.
Your presentation was really good. It's just we have fundamental question about the science behind it, which you cannot, I mean, if they don't say it, they don't say it.
But maybe we can try it out with the UMAP with like 3 dimensions. But
we should try it out.
Could it be that they use 2 dimensions because they only have 2? Well, they only, yeah, combining neural and behavioral labels, but not the time labels, exactly. And I think maybe the 3rd dimension that is added for the CEBRA is the time label. I don't know why they didn't add it to the UMAP. But
I think you still can choose how many dimensions you want it to be. I mean, as far as I remember, like you can put 3 dimensions, it doesn't matter how many variables.
Yeah. But for some reason they're really stressing that we can't include time information that is always given in neural recordings into other algorithms or into UMAP, for example.
Yeah, maybe there are some specific features that go against the rule of UMAP, so you cannot apply it, and then it will be not reliable. Maybe that's why. Yeah.
No, I say if you take your entire data set and you UMAP it, and then you display on the third dimension the time variation of the distribution of the points, boom, you did UMAP with time representation.
Yeah, but the spike at one time point and spike at the other time point, they are connected.
I see.
Yeah, maybe that's UMAP cannot do.
Oh, can I have like model the interaction of the same neuron across the
And also, well, the strength, what they also stress is that they can generate those embeddings. So this zebra, hold on, maybe. That it basically looks the same for a task. So for, yeah, it's basically in that figure, right? So that embedding looks exactly the same for, not exactly the same, but very, very similar for each rat. And they can do that across subjects, like across rats or rhesus monkeys or macaques they used, I think. And they can do that across experimental sessions where, for example, the distribution might be shifted across, experimental sessions or subjects or recording modalities. And this algorithm is apparently able to combine all of them and to still yield accurate accurate, consistent results.
We're gonna have to try it.
We can try it. The woman was here in Bordeaux and was and actually had a presentation here on that, and that's where I got hooked on it. And I was like, oh wow, that's cool. And there came also a question in the later in the discussion whether this can be applied with fMRI data, for example, of humans, and that could be actually extended. So
you should try the DSD on it.
Can we?
You should try the disconnectome.
Maybe that's the project for the second year.
Yeah.
But all right. Do you
Thank you.
Yeah, sure. That was lovely. Thank you for the discussion and for the questions. Yeah, sorry that we failed with YouTube. YouTube, see you in an hour when it's going to be uploaded. And thanks for joining. I hope you have an exciting week in science. Happy Monday. Bye-bye. Bye-bye.
Okay.